Okay, children, just the last topic was left from the previous chapter, that was the effect of pH on on the enzyme activity, right? So we share my screen. Okay, I believe you can see the screen now. So we know that the optimum pH for most enzyme is 7, that is neutral. But some enzymes are produced in acidic conditions such as stomach, so their pH could be pH 2, while some are produced in alkaline conditions such as duodenum, so their pH might be 8 or 9, right? If the pH is too low or too high, the bonds that hold those amino acid chains together that make up a protein, those bonds can be destroyed, and that can change the shape of the active site. So the substrate can no longer fit into it, and that is going to reduce the rate of activity. So that means that moving too far away from the pH, optimum pH, it will cause the enzymes to denature and activity will stop. See, here we had the enzyme, and this was the active site. So both of them were of complementary shapes, right? When the pH is increased far above the optimum value, so you could see that the enzymes active site change. So now this substrate can no longer fit into the active side of this enzyme. We can see this with the help of a graph as well. Here, this was the optimum pH, and this both ends are farther away from the optimum pH. So this area is the end at extreme pH. It is the farthest from the optimum pH. So enzymes, they are completely denatured, or you can have no activity over there. While moving away from the optimum pH will result in gradual loss of activity, as active site will become distorted, or the enzyme will be denatured. While this optimum pH, now the optimum pH depends the enzyme works in the body, like where the enzyme works in the body. As we have just studied that for stomach, enzymes that are present in stomach, the pH is 2, right? So that is the optimum pH for the enzymes that are present in stomach, while the enzymes that work in duodenum, their optimum pH is pH 8 or 9. Is that clear? Okay. The first question, okay. Okay, the first question is for Liba. Large biological molecules are made from smaller molecules. Which row shows the correct molecules? Liba, can you solve this question?
Yes, ma'am.
Okay, according to you, which is the best answer?
Ma'am, is it D?
Good. It's D. Okay, the next question is for Mibsam. The table below shows the elements and basic units used by the cells to make large biological molecule. Which row of the table is correct? Yes, Mibsam. What is your answer? A. Okay. Yes, Mibsam, you're right. The correct answer is A. Now the next question is for Laiba. This question is about biological molecules. Choose words or phrases from the list to complete the sentences. And each word or phrase may be used once or more than once or not at all. And here you have the plants. Yes, Laiba, can you start telling me?
Yes, ma'am.
Fats are composed of the elements.
Carbon, hydrogen, and oxygen.
Good. All proteins contain these three elements and the element.
Nitrogen.
Good. Glucose is a type of.
Carbohydrate.
Good. It's a type of carbohydrates. Animals store excess glucose as.
Glycogen.
Very good. Plants store excess glucose as.
Starch.
Good. And plants also convert glucose to dash.
Cellulose.
Very good, Libra. You're absolutely correct. Okay, next question is for Ms. Mipsam. Dash shows group of biological molecules and the building blocks that make them. Join the boxes with an appropriate manner using lines. Mipsam, can you try with your mic? Okay. Next question is for Laiba. Shows the components of fats and oils as shape. Using these shapes, sketch the appearance of glyceride molecule found in fats and oils. Okay. So as that I can do for you, as this was glycerol, right? Very good, ma'am Sam. You are telling absolutely correct. So this was glycerol, and then these were the fatty acids. So that is how you can actually combine them. Okay. Then describe the test that would be used to show a positive result for a presence of protein in a sample of food. Yes, Laiba, can you guess the test? Good. Yes, ma'am, Sam, you are correct. Yes, Laiba, can you make a guess for this?
Yes, ma'am.
Okay. What it could be?
Like add Biuret. That's the
Good. You have to drop some Biuret test solution into a sample of food, and then the positive result is a color change from blue to purple, or that is lilac, right? Okay. The next question is for Numan. Uh, okay. Numan, can you tell me that why the test for lipids is known as emulsion test? Yes, Numan, can you make any case related to this question? Okay. Numan is not in the class. So, as this question is for 2 marks, so we can state that A suspension of small droplets is formed. First, this reason, then we can say as lipids come out of solution because they are insoluble in water, right? And you can also state that the solution turns cloudy. That is also correct. Okay, children, that was for biological molecules. The rest of the questions you have to solve it by yourself. And if there's still any difficulty, you could consult me, right? Now we have to do the questions related to the chapter enzymes as we have completed this chapter. Okay, the question number 2 is for LIBA.
Mom, the answer is B.
LIBA, is that B or D?
B.
B. But is a feature of all, they are not changed by the reaction. Okay, that's correct. Okay, okay, perhaps I'm good. Then you have to tell me what is the correct description of the enzymes. Again, that is for Mepsam. Mepsam said so that the correct answer is B, that is biological catalyst. Okay, yes, Mepsam, you're also correct. Okay. Okay, the diagram below shows an amylase enzyme catalyzing the breakdown of the part of a starch molecule into smaller molecules of maltose. Which diagram has three correct labels? Yes, this is for LIBA.
Mom, can you please zoom in a bit?
Okay.
A.
So A is the correct answer. Good, Mepsam. So these were MCQs. Now we have to see some of the structural questions. Okay, figure 1 represents the reactants and products in a chemical reaction and four different enzymes A, B, C, D. State the letter of the enzyme responsible for this reaction. Mepsam said so. The correct answer is okay. Mepsam, according to you, the correct answer is okay. Mepsam, according to you, the correct answer is C. That is correct. Okay. Yes, Laiba, this question is for you. Okay, I have to write it down. what happens when a substrate with the shape that is not complementary to the enzyme tries to enter the active site. This question is for 2 marks, so you have to give two good reasons.
Mom, the substrate does not fit into the enzyme's active site, so it cannot bind properly.
Okay. So we can say that the substrate, you are also correct, Mipsam, very good. I appreciate that you are taking part in solving these worksheets. The substrate is not able to take part or is not able to bind efficiently to the active site. Other than that, as this is for 2 marks, so we can say that no reaction is catalyzed, or you can say no reaction proceeds or occurs. Okay, then we have another question. This question is for Mipsam, that the pancreas is an organ that produces enzymes used in digestion. Explain the importance of enzyme specificity in the digestion of food. This question carries 4 marks. Yes, Mipsam, digestion. Okay, enzyme. Okay, Mipsam, you said that the accurate reasons. Let me break down some for you. That we know that the enzymes, they are proteins and have a specific 3D shape. Other than that, enzymes have a specific specific shaped active site. Other than that, we know that the enzyme forms an enzyme substrate complex with a substrate, and the products of enzyme digestion or activity are absorbed through the small intestine, right? And one more thing we can mention is If the enzyme does not form an enzyme substrate complex, there are no products of digestion. Digestion to be absorbed, right? So these are the four reasons that you can mention. Okay, now we have to see. Okay, this question is for Laiba. Glutaminase is another enzyme that has shown success in treating cancer. It is within a group of enzymes called amino acid deprivation enzymes, which act on and destroy specific amino acids in cancerous cells. Explain the action of glutaminase uh, on amino acids in cancer cells. Yes, Laiba, can you hear me?
Yes, ma'am.
Okay.
Okay. So my first point is that glutaminase converts glutamine to glutamate. So this is a glutamase catalyzes the conversion of glutamine to glutamate and ammonia.
Okay.
And my second point is that cancer cells rely heavily on glutamine, and
Hmm.
many cancer cells are glutamine addicted. They require a large amount of glutamine for energy production and synthesis
Mhm.
of nucleotides. My third point is glutaminase disrupts cancer metabolism by converting
Okay.
glutamine to glutamate.
Okay. What else?
My fourth point is normal cells can survive with lower levels of glutamine or can use alternative pathways.
Okay. So actually, you have to explain the action of glutaminase on amino acids in cancer cells. However, your answer is also correct. We can alternative answer to that would be that glutaminase has an active site that is complementary to the amino acid in the amino acids or substrate that is in the cancer cell. Other than that, then we can say that glutaminase and substrate, glutaminase and the substrate or the amino acid form an enzyme substrate complex. Then the glutaminase, it catalyzes the reaction, and then we have the cancer cells. We know that the cancer, okay, They cannot con okay, you can write that the amino acids is no longer functional. They're no longer functional, or you can say cannot form proteins. And the cancer cells cannot continue to grow, right? Okay, then we have the questions related to enzyme action. Okay. So the pH of an enzyme controlled reaction is decreased by a pH of 2. How does this affect the rate of reaction? Yes, Sam, this question is from you, and your answer is that D. It may increase. Let me see. Okay. How does the affect the rate of reaction? It has no effect on, according to you, the correct answer. It may increase or decrease the rate. Okay. Okay. Yes, ma'am. Sam, you are correct. Okay. The next question is for Liva. Okay. Liver cells produce an enzyme called catalase, which breaks down the toxic hydrogen peroxide into water and oxygen. So a student set up an experiment where cubes of cow's liver in a test tube were incubated with hydrogen peroxide at different temperatures. The student counted the number of bubbles of oxygen released per minute at each temperature. Their results are shown in the table. So the results suggest that the optimum temperature for cattle is okay. Maybe Sam guessed it. Okay, Laiba, what is your answer?
Ma'am, is the answer B?
Yes, the correct answer is D actually. That is 20 degrees and 40 degrees centigrade. Because D has the highest number of bubbles recorded